So we have been mostly talking about linear messenger RNA all these days. In this lecture, we are looking at a different topology, circular RNA, which is a covalently closed loop, and that change in shape fundamentally alters how it behaves in the cell. Stability, translation mechanism, how it interacts with immune sensing and decay pathways, all are different. So that means the design rules change as well. For engineers thinking about sequence design or predictive modeling, features that matter for linear mRNA, cap dependent initiation or poly A tail dynamics, so those things don't apply the same way here. So we will walk through how that topology shift changes the biological constraints and what it means for engineering circular RNA systems. So as we just discussed, a circular RNA is structurally different from linear mRNA. Instead of a strand with a defined beginning and end, it is a loop. There are no free 5 prime or 3 prime ends. In cells, most circular RNAs arise through back splicing. A downstream splice donor joins to an upstream acceptor, producing a continuous circle. Because it's circular, the two hallmarks of linear mRNA, the 5' cap and poly A tail, they are absent. That has major consequences. How the RNA is degraded, how ribosomes initiate translation, how it interacts with cellular processing machinery, all these things change. So circular RNA is not a not a simply modified linear mRNA. It should be thought as a different architecture with the different design rules. So let's see why circular RNA is so stable. We discussed about linear mRNA that it has structural vulnerability because of exposed ends. Exonucleases recognize those ends and degrade the RNA from either direction. XRN1 from 5' end, exosome complex from 3' end. So that's why most linear mRNAs have half-lives on the order of hours. So circular RNA changes this at a structural level, and that is a closed loop. So no free ends. So exonucleases have no entry point. And as a result, circular RNAs are largely resistant to standard decay machinery, with half-lives extending to many hours or days. It's not indestructible, obviously. Endonucleases, for example, can still cleave internally. But the key idea is that stability here is driven by topology, the physical shape, not just sequence features. So as we just discussed, circular RNA gains stability by losing its ends. But that comes with a trade-off, that is translation efficiency. A linear mRNA initiates translation at the 5' cap. So the cap recruits, proteins like IF4, which assembles the initiation machinery and brings, pre-initiation complex. So this is highly efficient. It's why linear mRNA produces a strong burst of expression after delivery. But circular RNA has no 5' end. So cap-dependent initiation is not possible. Instead, translation starts internally through IRES elements, which let the ribosome bind within the sequence rather than scanning from 5' end. But this mechanism is less efficient, lower initiation rates, so the expression profile shifts. A linear mRNA gives you a high, fast peak, whereas a circular RNA gives you lower but more sustained output over time. Once we move to circular RNA, the sequence design requirements change as well. So first one is an internal translation element. So typically IARES. The choice of IRES is a major determinant of translation efficiency. Next, the coding region. Optimization follows similar principles to linear mRNA. Codon usage, translation kinetics, RNA folding, those things. But structure plays a bigger role here. The IRES depends on specific secondary structures to recruit the ribosome. At too much structure around it can block access. It's a balancing act. Finally, the molecule needs elements that enable circularization, the complementary flanking sequences or engineered systems. And in practice, circularization efficiency is often the main technical bottleneck. So producing circular RNA is a bit more involved than linear mRNA, but it starts the same way, with a linear transcript from IVT. From there, there are two main strategies. First, enzymatic circularization. An RNA ligase, like T4 RNA ligase, joins the 5' and 3' ends to close the loop. So the thing is that not every molecule circularizes, so you end up with a mix, and purification to remove leftover linear RNA becomes critical, obviously. Second, self-circularizing constructs. The RNA is engineered with ribozyme systems or complementary flanking sequences that drive circularization during or after transcription. This sidesteps ligation inefficiencies but adds sequence design constraints. But in both cases, the core challenge is, same thing, that is maximizing circularization yield while minimizing, contaminants. So circular RNA changes two things, immune recognition and stability. No free 5' end means no exposed triphosphate. So circular RNA sidesteps that class of innate immune sensing. And without a cap or poly A tail, the major decay pathways, deadenylation, decapping, they cannot initiate. So that's a big part of why circular RNA persists longer. But it's not invisible to the immune system. Double-stranded RNA byproducts from manufacturing unmodified nucleotides, they trigger TLR7, 8, or RIG1, viral IRES elements that resemble pathogen motifs. All of these can still activate innate sensors. So circular RNA reduces immune risk, but careful design and purification remain essential. Although circular RNA is promising, several technical challenges remain. Circularization efficiency is still a problem. So you get mixtures that require careful purification. And removing those linear contaminants matters because they can trigger immune activation. IRES performance is highly context dependent. It varies across cell types and structural contexts, making it hard to predict translation efficiency. And predictive models for circular RNA expression are still limited. We don't yet have reliable computational tools to forecast how a given design will perform. Finally, the basic translation dynamics, how ribosomes load onto a circular template, how they recycle, are still not fully understood. So circular RNA is an exciting platform, obviously, but much of the design space remains empirical. A circular RNA changes the core assumptions from linear mRNA. No cap, no poly-A tail, and topology becomes the dominant stability determinant. For a predictive modeling that shifts the focus, IRES performance, structural context around initiation, circularization determinants, and long-term stability pathways, all those things. The platform is promising, but data sets are not that many, and assays are not super standardized, which means there is a significant opportunity for modeling and ML.